So far, this talk has focused on PCR testing, which is considered the gold standard test, as it is the most sensitive and reliable test for detecting current infection. However, the test needs to be performed by trained laboratory staff using expensive laboratory equipment, and it takes time for the test result to be available. There are alternative tests for COVID-19, and in certain situations, these can be more useful. We will explore these.
Antigen lateral flow testing is widely used for community testing and was introduced initially in schools and the healthcare sector, but is now available to the public. Sometimes known as a rapid test, results are produced within 30 minutes. Similar to the PCR test, a person takes a swab and swirls it in a small tube containing an extraction buffer to release any virus present into the buffer. From this small tube, 2 drops of buffer are loaded onto a pad in the sample loading window of the lateral flow device.
The aim of this test is to detect internal proteins of the virus, which we refer to as the antigen. We use antibodies to detect specific antigens, and in lateral flow testing, when extraction buffer is added to the sample pad, it moves along the device by capillary flow, enabling the antigens in the sample to come into contact with the antibodies. If the viral antigens are present in the sample, then they will first bind to antibodies in the conjugation pad, which are labelled with a dye. There is another protein in the conjugation pad, seen here in purple, which is also labelled with a dye, which acts as a control for the device to make sure it has worked properly. The labelled antigen-antibody complexes flow along the device and bind with further antibodies at the test line to give a visual positive result after 20 to 30 minutes. As the sample continues to move across the device, the control protein labelled with a dye binds with antibodies at the control line. If a line does not appear at the control line, the test is a void result, no matter what appears at the test line, and must be repeated. Here's an example of a lateral flow device showing a positive result for the detection of COVID-19. Antigen lateral flow tests are less sensitive than PCR, and positive test results need to be confirmed by PCR testing. However, they are cheap, easy to use, and give results in minutes, making them ideal for large-scale community testing in people that have no symptoms of COVID-19.
So we've looked at testing for current infection. Now let's look at how we can detect past infection. This is known as antibody testing.
When we have an infection like COVID-19, our bodies produce antibodies, and these remain in our blood for some time. We can test for these antibodies to find out whether someone has previously had COVID-19, even though they may be fully recovered. Understanding past infection is useful because it helps us to understand where and how the virus is spreading in the population.
Antibody testing to detect past infection is performed with a blood sample as opposed to a nasal or throat swab. To detect COVID-19 antibodies, we use a biochemical test called an immunoassay. These tests use antibodies themselves to detect and measure specific molecules, in this case the COVID antibodies. Antibodies are really useful in laboratory tests because they will only bind to specific proteins or antigens or specific antibodies, and we can attach molecules to them so that we can then detect and measure them. Enzymes are often attached to the antibodies in immunoassays, which are really useful as they help generate a light signal that we can measure. These types of immunoassays are known as enzyme-linked immunosorbent assays, or ELISA for short. Let's have a look at an example of an ELISA, which is used to detect COVID antibodies. This ELISA is carried out in 96 well plates, which allows for over 80 test samples plus control samples to be analysed at the same time in individual sample wells on the plate. The bottom of each sample well of the plate is coated with the virus protein. The first step of the test is to add a blood sample. If the sample has COVID antibodies, then they will bind to the virus protein. Next, we add the detection antibody, which will bind to the COVID antibodies if there are any present. The detection antibody has an enzyme bound to it. And when a specific chemical known as an enzyme substrate is added, a colorimetric reaction occurs, which means the sample well changes colour. Sample wells with no COVID antibodies do not change colour. You can see colour changes in the plate on the left. A strong colour change would indicate a lot of COVID antibody is present.
ELISAs are sensitive and reliable. However, like PCR testing, the test needs to be performed by trained laboratory staff using expensive laboratory equipment, and it takes time for the test result to be available. An alternative to the laboratory test is to use a lateral flow test.
The antibody lateral flow test works in a similar way to the antigen lateral flow test, except here you're using a blood sample from a finger prick as opposed to a nasal and throat swab, and you are detecting COVID antibodies to show past infection. It takes 1 to 3 weeks to detect antibodies after COVID infection, so antibody tests are not used to confirm the presence of active virus. All you have to do for the antibody lateral flow test is to add a drop of blood onto the flow device and 2 drops of buffer. Let's have a look at a positive test result.
There are 3 lines in the test window. The control line must appear or the test is void and must be repeated. A positive result on the G line indicates that the person has IgG antibodies, and a positive result on the M line indicates IgM antibodies. IgM antibodies may indicate a recent infection as they decay more quickly than IgG, which can hang around for several months after an infection. Antibody lateral flow tests are less sensitive than ELISA. However, they are cheap, easy to use and give results in minutes, making them ideal for large-scale community testing to enable scientists to understand and model how COVID disease has spread within populations. This is important to guide further control measures to stop the spread of disease.
Like all viruses, the COVID-19 virus can change or mutate when it makes copies of itself. 4 main variants have been identified, which the World Health Organization have labelled Alpha, Beta, Gamma and Delta. You may know them as the UK, South Africa, Brazil and India variants.